Hello students, how are you people? Hope you're all doing good. Well, once again welcome back to our biology class. Myself Dr. Raghavendra here. In the earlier class, we had done a discussion related with the important concepts under the chapter Principles of Inheritance. Now, the next chapter Molecular Basis of Inheritance, and in this some of the important concepts which are very closely associated with the human genome project as such, as well as we are going to discuss one more important concept called as the DNA fingerprinting. In today's class, we will have the discussion, we will have the study about the human genome project. In short, it is also called as HGP, right? So human genome project. What are the important concepts? Yes, we will be discussing under this. Number one, we will study about a few introductory points related with the Human Genome Project introduction. Number two, we will discuss about this project is also called as mega project. So why so mega project it is called as? We will discuss about this. Number three, we will discuss about what is the intention of this project, the goals of HGP we will discuss. Number 4, we will discuss about what is the history behind this, when it got started, when it is finished, history of HGP. Number 5, we will discuss about mainly the application part, applications related with human genome project. Number 6, we need to understand what are the important and different methodologies are utilized. So we will study about mainly methodologies. When so methodologies we will discuss. Number 7, salient features of HGP. Salient features of human genome project. Number 8, we will discuss about what are the major challenges. Yes, mainly related with human genome project. So these are the important concepts or aspects related with this important topic, what we call as a human genome project. So what do you mean by a genome? Whenever we just ask a question, can you define what is a genome? Genome is basically, total number of genes. We can say that definition for the genome, it is total number of genes on haploid set of chromosomes. Means human is being deployed. Yes, we can say human is 2n. That means human has 2 genomes. So there are 2 genomes present in the human. So generally the females, what is the total number of chromosomes in the human? Yes, we have 46 chromosomes. Okay, 46 chromosomes. In that, we can say that females have 22 autosomes, plus again X chromosome. This is the one genome. Another genome, 22 again autosomes plus X chromosome. Both the 2 genomes in the female are same. So they have 2 identical. So they have 2 identical genomes. 2 identical genome. Means in the female, if you study one genome, that's enough. No need to study another genome. But whereas the male has 22 autosomes plus X chromosome, 22 autosomes plus Y chromosomes. So males have 2 non-identical. There is a difference because of the X and Y. So 2 different. Now two different genomes because of due to X and Y chromosomes. This you need to remember. Ultimately, what do you mean by the genes as such? So, when we talk about the genes, you may be knowing they are not the clothes what we wear. Genes refers to. As such, we say that here is the cell and the cell is having nucleus. Okay, nucleus, this is the cell. But nucleus can't be empty. It has lots of this chromatin material. Chromatin. So, or the chromatin is well packed inside. What is this? We say chromosome. So, in the chromosome, yes, there is lots of packed material, and what we find, it is the DNA. And on the DNA, yes, we can say this is the DNA. So here it can be 5' and 3', here 5' and 3'. There is adenine pair with thymine, guanine pair with cytosine, like this. Lots of again cytosine, again guanine, again cytosine. So these AGGCC, what we say, sequences. So the sequences, we call them the sequences, also called as the genes. So genes are the part of DNA. They are the sequences of the DNA. They are the polynucleotide, we can say, yes, they are the long chain of nucleotide bases. Then we call them as mainly the genes. So understanding how many variety of genes that exist inside the human body, and then what is the role of that gene, so how they will influence phenotype as well as the further metabolism of the body, all that, yes, what we say, that is the ultimate intention of mainly the Human Genome Project. So let me tell you here, somewhere in 1970, there was a person by name Walter Gilbert. Walter Gilbert developed a technique. What is that technique called as electrophoresis? So electrophoresis, sometimes it is referred as gel electrophoresis. What is the use of this gel electrophoresis? It is the most important technique that is helpful to separate the biomolecules. Yes, the biomolecules, let it be a DNA or RNA or proteins, that can be separated in the porous gel under the influence of electric field. So ultimately electrophoresis is a technique separation of biomolecules in a porous gel under the influence of electric field based on their size or molecular weight. So mainly the biomolecules can be separated. Let me give you a definition of this. Electrophoresis is a technique to separate biomolecules in porous gel under the influence of electric field. Okay, so the biomolecules get separated. Molecules get separated based on charge and molecular weight. So what happens here? Yes, you know, DNA is it is a negatively charged and it will move towards the positive electrode, and the DNA, when they are very small fragments, they move very fast. In the gel electrophoresis, yes, you might have been studied that there will be a gel sheet like this, and then yes, you will find like this, they will make what we can say the micro wells, or the well in which they will add the DNA, and then So here it is a negative electrode and here it is a positive electrode, and this all this set kept. It is a gel sheet. Okay, it is a gel sheet which is porous by nature, and here what we added is all the DNA samples. Then DNA samples start getting moving up, moving down. So the larger molecular weight DNA will stay there, and then yes, so the smaller molecular weight DNA will move downwards. So thus, in fact, it's all the buffer tank. What is filled here? It's all the buffer, and then yes, we say buffer tank. So thus DNA gets separated. Mixture of DNA with a different molecular weight, different size. When they get separated, these small bands we can cut and we can isolate the required DNA of a specific molecular weight. This is the most important and foundation technique for the furthermore development of entire human genome project. This kind of technique is given by Walter Gilbert in 1970, right? So with this technique, yes, it has become the basis for the further development of the human genome project. And then somewhere in the 80s, in 1980, yes, there was a person, yes, mainly a company by name. The CEO, the name of that CEO is mainly Craig Venter. So CEO Craig Venter of Celera Genomics, okay, gave, yes, proposal of human genome project. He said that yes, I will be mainly isolating the genes, I will be separating the genes, and then I will decode the entire human DNA, and then his intention was to sell that gene information to the companies, mainly the pharma companies, and they will develop a particular kind of drugs or medicines for the recorrection of defective genes. So this kind of idea and proposal, Craig Venter came in 1980. But in 1990, US government took initiation, and yes, they did not give to the private company, as the technique may be or the information may be exploited by the number of MNC multinational companies, and then there will be monopoly on the information, and people who need the drug or the medicine for a particular gene defect, then they will not be able to get it. So to avoid such consequences, yes, US government decided to start the human genome project on its own, and the first director of HGP, yes, was James Watson. So it started in 1990 and HGP completed in 2003. Well, so James Watson is the same person who gave, along with the Crick. So Watson and Crick, they are the people who gave double helix model of the DNA in somewhere in 1950s, and they got the Nobel Prize in 1962. Well, so the same person, yes, become the first director of this human genome project, and then it just started. Well, so this is a brief history what we have seen, or little bit of introductory point related with the human genome project. So we need to get into the details, in fact, related with the human genome project. Why this project is called as a mega project? Let us go ahead now. So let us understand why this project is yes, considered to be as a mega project. I hope you got an idea. Genome refers to the total number of genes. As such, earlier people have mainly imagined there may be lakhs of genes present in the human genome, but not a true. Yes, hardly 30,000 genes have been identified. Well, so let us understand why this project is known as a mainly what we say mega project. So what are the reasons why this mainly the human genome project, it has been named as a mega project? So there are lots of information, yes, we need to know. So definitely it is a mega project because it is not a simple work to mainly isolate billions of genes or billions of bases that existed in the human genome. And then studying of all those genes is not a simple task. That's why, yes, it has to be considered as a mega project. HGP as a mega project. As a mega project. Why? Because the reasons are simple. Number one, entire human genome has approximately 3 into 10 raised to 9 base pairs. These many huge number of bases, what we can say the base pairs means adenine, thymine, guanine, cytosine, like this. We call them as a base pairs. The huge number of base pairs do exist and mainly sequencing them is not a simple task. That's why it is considered as a mega project. Number two, so the estimated cost for sequencing single base pair is $3. Thus it may cost around 9 billion US dollars. That's why it is a high costing, what we can say, project as such. Number 3, so highly skilled technicians, scientists, sophisticated, what we can say, the devices and the, what we can say, yes, scientists, sophisticated device, and then high-tech labs required. That's why, yes, it has been to be considered as a mega project. Other than that, as I told here, 3 into 10 raised to 9, yes, nearly, maybe we can say that 3 billions of base pairs have to be studied well, analyzed properly, and then it has to be sequenced well. All that work requires more, what we can say, time, more investment, sophisticated labs, the what we can say, high-tech laboratories we can say, and then yes, the devices, advanced instrumentation is required, and then people are required, right? So thus even the funds are required enormously. That's why it is considered to be as a mega project. And if the sequences have been keep on writing on a paper, how much time it needs? It may need more time, as well as we can say that if the sequences have written on the paper, the fourth important point, if the sequences have to be written on paper, so minimum 1000 letters per page, 1000 letters per page, like that 1000 pages per book, like that 3300 books, okay, required to write the sequences per genome, okay, from a single cell. So this much we can say that investment of time, energy, technicians, scientists, money, devices, all that needed. That's why this human genome project is considered as mainly a mega project, so which is not at all a simple task. People also say that, for instance, give the example, if you keep on writing all the letters on a paper or typing on a paper, minimum 1000 letters should be there on a page, and like that 1000 pages in each notebook, like that 3300 books are required to write the only sequences of one genome of a single cell. Okay, so thus the human genome project is going to become a mega project. It's a large project, right? So, and people also say that if you keep on piling up all these information one above the other, or notebook one above the other, it may, yes, may cross the height of Eiffel Tower also, they say. So just a simple instance of the example what I suggested. So thus human genome project is the cost effective, laborious, technicians, scientists required, even more number of things or time investment also needed. That's why it is called as a mega project. Well, let us go ahead. What are the then goals of this human genome project? So, when we talk about mainly the goals, HGP, yes, has number of important goals, and that can be most useful for the mankind as well as to conserve the nature also, we can say. So human genome project, yes, it is the involvement of number of people, and human genome project goals are very essential and important. HGP, yes, the goals. What are the important goals? Number one, so here sequencing entire human genome. That means approximately 3 into 10 raised to 9 base pairs to be studied well. Okay. Number two, yes, sequencing the entire human genome information, and then storage of HGP. Okay, sorry, the storage of the entire sequence information. Entire sequence information is also called as a database. Database in computers. So that any time the sequence of chromosome 1 to, yes, 46, yes, any time, any chromosome, any gene information, we have to be able to retrieve it. So take it out and utilize it. Otherwise, writing on the paper and then, yes, it is highly laborious and a difficult task. So storage of the entire human, the genome information or the sequences as such 3 to 10 raised to 9 base pair sequences. Number three, so improvement of technologies, okay, to carry over HGP. So with the conventional methods it was very highly difficult to do the sequencing. So improvement of the technologies to carry over the human genome project. Number four, yes, so here the information or database, okay, transfer as per requirement for the benefit of the mankind, okay, and then, yes, so number 5, here mainly what we can say that the sequence of the entire genome, storage of the entire sequence information, improvement of the technologies, information or data transfer, even tools and technology transfer also one of the important goal of the HGP. Other than that, addressing when human genome project releases out all the sequence information, those sequence information can be misused by the number of multinational companies. They may develop what we can say their own kind of softwares, their own kind of what we can say the particular gene related medicines. So in fact the tools and technologies to transfer, and it has given out mainly when they have stored lots of information of the gene sequence or database. So it gave a the entry of or development of the new field, what we can say bioinformatics. Bioinformatics. So whenever the information is required related with the biological, yes, mainly organisms or the gene, yes, that can be retrieved, and the field is called bioinformatics, has emerged out when they collected huge amount of data or information related to the human DNA. So now here, the sixth and most important goal to address the ethical, legal and social issues. So what are the ethical, legal and social issues? Once the Human Genome Project give all its information about the human related genes, there is a furthermore development of the transgenic plants and animals, and then highly nutritious or the therapeutic important medicines can be developed from the either plant material itself or maybe the animal material itself. Maybe in the milk or in the egg we can have the human proteins which are medicinally important proteins. So all such things will develop, and this also will have a certain social impact. People have a misconception or wrong assumption that there may be a problem with this kind of the hyper or high nutritious food. They may think like that. So number of ethical and legal issues, yes, people mainly all over the world will try to exploit the human genome information, and they develop the medicines related with the target medicines for the what we can say the mainly the gene defect to give the remedy for that. The field is called as the pharmacogenomics. So MNCs with the pharmacogenomics, they may play big role and they may exploit the world medicinal requirement. So the ethical, legal and social issues also to be addressed, that with the human genome project such illegal things will not happen, such abnormality will not happen. Human genome project is mainly for the benefit of the mankind, that has to be addressed. This is the main goal of the human genome project. So legal issues as such, as I told you, the HGP information yes further can be helpful to produce gene targeted medicines, and that field is called as pharmacogenomics. So two important fields have emerged out. One is called as bioinformatics, other one is pharmacogenomics, but the information has to be utilized for the betterment of the mankind, not to exploit the human. So this is all what we say mainly the goals of a human genome project. Well, so in the next class, yes, we are going to discuss about regarding the history, applications as well as methodologies also. Till then, take care and study well. Thank you very much.